So first of all, I'd like to thank the organizers for this wonderful meeting. And today I'm going to talk on mechanical stability of bipolar spindle assembly. And this work is done in collaboration with Paulo, who is at Max Planck Institute for Intelligent System in Stuttgart. So first let us look at the process of mitotic cell division. So cell division, during the process of cell division, basically there is replication of genomic materials and there is segregation of the copies in the daughter cell. And this process of cell division is mediated by this structure, what is called spindle. So this is the fluorescently labeled image of a spindle. What you see here in green are the fluorescently labeled microtubules. So they form, so what you see is that you have these two centrosomes and these microtubule structures, they overlap. And the chromosomes are aligned during the formation of spindles in this midzone between these two centrosomes. And this process is mediated. So what the question is that we would like to understand is, what gives rise to the stability of the spindle-like structures which are formed? And in fact, during the process of cell division, you have metaphase. So this is one of the cycles of the cell division. And this spindle-like structure typically is stable for around 45 minutes. And eventually, of course, these two daughter cells would be formed. And so the various, of course, various complex interactions takes place. But in particular, these microtubules interact with the chromosomes, which are situated here. These overlapping microtubules also interact with the motor proteins. So in the last talk, what you heard was the roles that the motors played in leading to transport within the cell. So here what we see is that these motors also play a role in determining the structural stability of these kind of structures. And apart from that, there are also other cellular structures like kinetochore, which are also involved in the process of formation of the spindle and determining the stability. And the question that we want to understand is, what determines the stability of this spindle structure? So let us look at more a schematic of this, what is going on. So here you have these two centrosomes from which these microtubules polymerize, and then you have, so some of them interact with these chromosomes, which are denoted here. Some of these microtubules overlap, and these motor proteins, they basically cross-link these microtubules, and then depending on what kind of motors are there, they can exert an effective repulsive force between these two centrosomes. So if it's a kinesin motor, they will try to slide outwards. If there is dynein motors, they will try to slide inwards. In fact, recently it has been observed that apart from this cross-linking motors, there can also be other passive proteins which can be involved in the process of exerting forces between these microtubules. And some of them, like I mentioned earlier, these microtubules are also joined, these overlapping microtubules, by kinetochores. And lastly, there are also set of microtubules which reach the cell body, and there are specific motors, the cortical motor proteins with which they interact at the cell cortex. So these are sort of the, the different interactions or different interactions which are at play. And let us first now try to look at a more sort of toy model that, okay, if one wants to, quantify these interactions, let us assume that these microtubules are nucleating from the centrosomes, and they have some exponentially distribution of length with some mean length. And then the force due to these microtubules which are interacting with chromosome arms are assumed to be just proportional to the number of microtubules reaching the chromosomes, and we are taking that the separation distance between the two centrosomes is 2x. And then the force due to the interaction of these microtubules with this cross-linking motor proteins is taken to be proportional to the overlap length. Of course, this kind of description is in some sense an assumption. It's a kind of mean field description. And then finally, we assume that the force due to the microtubule kinetochore interaction is just some constant. And apart from that, you can also look at the interaction of this astral microtubule with the cell cortex. So this is for the case of where you have just two centrosomes and just one chromosome pair which is there. And then one can in fact write down a kind of expression for the force as a function of the separation distance between these two centrosomes, which has been discussed previously. The main conclusion of the study is that relatively, if you look at these four interaction terms, if the cell is large enough, then the interaction of the microtubules with the cell cortex is relatively unimportant as compared to the other three interactions that are at play. Now, this model has been studied or extended rather to study for the case of the general case that where all these chromosomes are there and these microtubules are interacting with these chromosomes and there is interaction which is arising due to these motors. And how it has been modeled is that so Ri is basically, if you consider n chromosomes which are distributed, then Ri is the separation of this one of the centrosome from each of the chromosome, and then you have to sum over all the interactions for all these n chromosomes, and then one can write down an expression for the force which is acting between these two centrosomes. And then finally, in this numerical study, one has to also model how the chromosomes interact between themselves, and that is modeled through some short-range interaction which is linear in the separation. And the technique is that you write down basically the equations of motion for the interaction of these n chromosomes, and you solve these equations in the overdamped limit for various different initial conditions. And the main sort of key results that was found was that of course the relative strength of these interactions determine when you will get this kind of spindle, when this kind of bipolar spindle would be stable, and under what parameters you don't see the formation of this bipolar spindle. And it was also noted that the spindle configuration also seemed to be sensitive to the initial condition. So if you take what is the initial separation between the two centrosomes that you start off with, that also seems to play a part. Finally, one of the results that I would like to focus on, because that would be the starting point, is that the chromosomes in the steady state were seen to be distributed in the mid zone between these two centrosomes, and they were either in a sort of disk configuration along the mid zone, which was perpendicular between these two centrosomes, or they were distributed on a ring configuration. Now, this nice feature, we will sort of say that, okay, we will assume that we will not look at the full dynamics, but rather we would like to quantify what would be, we'll assume the configuration that these chromosomes are distributed on this ring configuration. And then the nice thing is that one can actually write down a closed form expression for the force. Because because due to the symmetry of the problem, the force along the y direction, the other direction, perpendicular direction would cancel out, and one can write down the forces even for this configuration. One just has to do the integral, and then one can actually go to a more, one can define scaling variables and have this kind of closed form expression for the force in terms of the scaling variable. Note here, this B naught and C naught, this characterize the strength of the motor interaction. So B naught will be positive if it's dynein motor which are exerting a net inward force. It would be negative when, or repulsive when it would correspond to a situation where you have kinesin motors. And finally, this C naught characterizes basically the strength of kinetochore interaction with respect to the force which is exerted by these chromosomes with microtubules. So once you have this expression for force, then one can actually associate an effective potential energy or dimensionless potential energy in terms of these scaling variables. Now we ask the question that if we are to look at under what conditions you will have a stable spindle configuration, and it's just basically the condition for mechanical stability just boils down to the forces the net forces going to 0 and the derivative being less than 1. And so if one wants to determine the stability boundary, then all that one needs to do is just equate it to 0 and set this to 0 to determine, that would be the condition for determining the entire stability behavior of this kind of configuration. And what we find, so we first do the stability diagram for chromosomes on a disk. What you see is that, so these two axes, you remember this axis sort of quantifies the relative strength of the kinetochore interaction, and this axis corresponds to the interaction due to the motor proteins with the centrosome. So positive B would mean more outward forces, whereas negative B would mean a net inward force due to the motors. And here you can actually see that or if you have a typically a positive value of B naught, then that corresponds to a situation where your bipolar spindle would be stable. If you go to more attractive interaction of B naught, then that corresponds to a situation where this bipolar spindle will not be stable. So you form a solution where the monopolar spindle would be stable, and this can be seen, one can actually plot, the expression for the effective potential energy, which basically characterizes that if it's a monopolar spindle, that would correspond to a situation like this, where the minima of the potential energy corresponds to a minima. Whereas you can also have this interesting scenario that for certain regime of parameter space, you can have a situation where there is two different solutions of spindle are stable. That means for two different separation length, you can have a solution which are bistable. So this would correspond to a situation like this, where you have two minima of the potential energy that would correspond to a region here. There can be other situations where you have one minima, even the monopolar spindle, which is just also stable, and the bipolar spindle is also stable. So similar thing can also be done for chromosomes on a ring, distributed on a ring, and do this kind of, you go through this entire process, and then one can find out in general the stability diagram even for this. And then we made some comparison with the computational model, which was much more involved. So the basic message is that it captures many of the aspects of this computational model. Of course, the quantitative details are slightly different. And the summary is that, this assumption allows us to find out the stability diagram and to quantify when you are going to have a stable spindle and when you are not going to have a stable spindle, depending on the strength of the interactions, which can be tuned in different experiments. And in particular, we also see that for certain regime of parameters, you also get this bistable kind of behavior with multiple minima in the potential energy landscape. Further, the fact that you have this multiple minima also seems to correlate with these numerical findings that it's sensitive to the initial condition. So the multiple minima in the potential energy landscape means that if you started in a region of separation where you had one kind of solution that would give to a certain kind of spindle solution with a certain length, whereas if you started in with some other initial conditions, you will have the other kind of solution. So with that, I would like to conclude my talk, and thanks for your attention.
Sudipto, is bistability seen in experiments? Positional bistability?
Yes. Meaning what in the bistability in the experiments?
Yeah, experiments.
The numerical simulations they see.
Any experiments showed such
I don't know
behaviors?
specific, but if I understand it correctly, they do see different kind of structure. Meaning you see different, whether it is easy to quantify and ascribe it to bistability in the sense that you see solutions of different spindle separations.
Solutions, I understand.
In the sense that you can have,
so the question I'm asking is that sort of do you even, yeah, but if you are in text like statistics, so do you see?
So I don't know if there is a quantitative, statistical analysis of that. What they do see is that, you start with this configuration, then they see a distribution of different, spindles of different separation. Same cell. And also there are these issues, in fact, the actual situation is more complicated. They sometimes also see this misaligned spindle. So there is a tripolar spindle, sometimes which are also. In fact, we are trying to understand what are the conditions when instead of a bipolar spindle, you have this tripolar spindle which is formed, then it stays. Sometimes it happens, some of them merge and form a. But I don't know of quantitative comparison of that. In numerics, it is.
Both kinesins and dyneins are pulling the microtubules, is it?
No, one is, so the kinesin is, because this is the plus end, imagine. So the kinesin are going to slide, so it's a net outward. If you look from the perspective of these two centrosomes, it's going to an effective repulsive force. Whereas dynein is like a effective, attractive force. In fact, dynein typically tends to destabilize the bipolar spindle configuration. In fact, the role of, apart from dynein, like I said, that recently people have also seen that the role of many of these passive cross-linkers can exert entropic forces, and they also play a role in this determining system.
Okay, if there are no more questions, then let's move to the next speaker. Let's thank Sudipto once more.